Hi everybody, it's Miss P, and here is this week's video about DNA mutations. So this is the last content we're going to cover before you start to study for your quiz. I have your quiz due next week, Monday, April 27th at 8 a.m. That means you have to have it done by April 27th at 8 a.m. Okay, so you'll have the rest of the week to work on that. Here's the module for this week in case you haven't been on Canvas yet. This is just for questions, as it always is. This is for learning goals, where you can check out what I'm expecting you to be able to do this week, what I'm expecting you to learn this week, and here are some tasks for you to do. You obviously found the mutation video right here. Study guide, and there's some other test prep materials in task 2 that you can use. None of them have to be turned in. They're all up to you to use to study the best you can for this quiz. The quiz is right here, and it's going to look is right here, I should say. And it is going to look pretty much just like any other quiz that we've taken in class. It's just going to be set up in Canvas. So you'll take the quiz in Canvas and you'll submit it on Canvas. We'll grade it on Canvas and we'll move those grades into Infinite Campus where they'll actually show up in your gradebook. After you take your quiz, I want you to do a little bit of a check-in for me. I want to know how you're feeling so far with this whole distance learning thing. You've been through 4 weeks now. You'll have finished an entire unit basically online and you'll have taken a test. So I want to know how things are going for you. Do you feel okay still? What can we do to help you better? There's a few questions in there to guide you as you make those decisions and make a response for that discussion. All right. So we've talked about mutations before, right? We talked about mutations when we studied the cell cycle, and we kind of came to the conclusion that most DNA mutations cause cancer, right? I saw that a lot in your guys' response to like, oh, if something goes wrong in DNA replication, what is that going to cause? And a lot of you went straight to cancer. But DNA mutations are about much more than cancer. So yes, they can lead to cancer. But DNA mutations are also a great way for us to naturally get some diversity in the world. DNA mutations are what made selective breeding possible. So if you have a lovely pet dog or a pet cat or another pet of any kind at home, that animal has been selectively bred over hundreds of generations to come from a common ancestor. For dogs, it's the gray wolf, into hundreds of thousands of different breeds of dogs that all look very different. And that's all because their genes and their DNA had mutated slightly. So we wouldn't even have any of that without DNA mutations. DNA mutations are also used in agriculture a lot and science a lot to make things like GMOs. So there's a lot of ethical questions about GMOs, genetically modified organisms, and genetically modified food. But basically, they only exist because DNA has the possibility of mutating. So what are a few types of DNA mutations? We're going to look at 5 types of DNA mutations. There's also an organizer on Canvas for you to take some notes about these in there. Basically what I'm looking for is the name of the type of mutation, a little description of it, and then like a picture or an example of what that would look like for the DNA. So our first type of mutation is called a deletion. This is when one nucleotide gets left out when DNA is replicated. So for example, over here we have our original DNA sequence, and when this DNA sequence got replicated, this nucleotide got left out. It got booted out, it didn't get copied, it didn't get added in. For some reason, DNA polymerase didn't put that thymine where it was supposed to go. Because of that deletion, all of the codons from this point forward in the DNA are going to be different than they were before. So remember, a codon is 3 nucleotides together. So 3 nucleotides equal 1 codon. So if this was a codon, it's now going to be different because this T is missing. It's going to shift everything over. We call that shift a frame shift because it shifts the codons that determine the amino acids coded for by this DNA sequence. We call that a frame shift. Another type of DNA mutation is called an insertion. So remember, a deletion is when a nucleotide was left out. Insertions are when nucleotides are added in as DNA replicates. So once again, we have our original sequence here. And right here between this C and this T during DNA replication, something happened, a mistake of some sort happened, and we ended up with an extra cytosine in here. Again, because we've inserted an extra nucleotide, that also made a frame shift. It shifted all of the codons in this case to the right side by one nucleotide, thus changing all of the other codons from this point forward. So if you look up here, we would have had TAA, CTG, CAG, but now we have TAA, CCT, GCA. It's a little different. A third type of DNA mutation is a substitution. This is when one or more nucleotides are replaced by the same number of nucleotides. So we're not technically losing nucleotides and we're not gaining nucleotides. We're replacing some nucleotides, one or many, with the same number of nucleotides. So for example, right here, when DNA was replicated, this codon right here, the TGC, I know it's a codon because it's 3 base pairs long. This TCG codon got kicked out and a GAT substitute got brought in. The DNA is still the same length. It didn't cause any frame shift because we inserted exactly what we took out. But substitutions can still affect the code for amino acids. So if you had an amino acid chart out right now, I'm not going to bring one up, but if you had one out, you could look and see if TGC and GAT would eventually code for the same amino acid. You would have to convert this to mRNA first, but you could tell. In this case, it will change the amino acid that this specific region codes for, meaning that a different protein is going to be made for the end result, and it's going to work a little differently than the original protein might have worked. This is how you get different alleles for your genes. So remember, alleles are like different versions of a gene. So there's like an allele for red hair and an allele for blonde hair and an allele for black hair. Maybe this change created a new allele where it's kind of a mixture of black and brown. I don't know. All right, the next type of DNA mutation we're going to talk about is called an inversion. So remember, when we invert something, we kind of reverse it. We flip it on its head, if you will. So an inversion is when a segment of DNA gets reversed from one end to another. So for example, the T was over here in the original sequence, but now that it's been flipped around, the T is over here. So we took this whole sequence and we flipped it on its head. We reversed the direction of this specific grouping of nucleotides. Once again, because we didn't technically lose nucleotides overall and we didn't gain nucleotides overall, this isn't going to cause a frame shift. Any codons before this part will still code normally, and any codons after this part will still code like they used to. But whatever codons were in this region specifically might be different because they're going to be read in the reverse of what they originally were. So this is probably going to affect that code for amino acids, which means it's going to affect the end protein, which means it might cause more genetic diversity. Not necessarily a bad thing. All right, and one more type of DNA mutation is called a translocation. So this is when a stretch of DNA on a chromosome is moved to another location on a different chromosome. We've talked about something like this before. For instance, if we take the end of one chromosome and we move it to another chromosome. No genetic material is getting lost or gained. All of those nucleotides inside this chromosome on this DNA strand are still being retained. So remember, this is a chromosome. Inside, there's DNA coils that make up the chromosome. So all of the DNA in there is still there. It's still in the same organism. It's still going to be coded for in the normal way. So you're not losing or gaining anything. Those genes are still going to be coded for in the correct way. They're still going to make the same kinds of proteins. So you're not actually losing anything. It's just moving things to different places. And we've heard about this before, right? We've talked about this earlier. This should look pretty familiar to you guys. Remember, during meiosis, we talked about ways to increase genetic diversity? This is exactly what we were talking about when we talked about crossing over. We were talking about the translocation of a chunk of a chromosome to another chromosome. Now, these chromosomes right here are not the same size, right? They're probably not homologous chromosomes, so they're probably not an this isn't a specific image of what crossing over would look like. But nonetheless, this is another way that we make genetic diversity without actually losing genetic material or changing it or risking those mutations that could cause cancer. So those were the 5 types of DNA mutations. One note about mutations, though. They're not necessarily always a bad thing, right? We talked about earlier, they're not just about cancer. So cancer would be something bad that came from a mutation, but there are also good things that come from mutations. Mutations increase genetic diversity, just like we talked about with crossing over. And sometimes, for example, in the case of substitution, they might not affect anything at all. This could substitute and bring in a set of a codon, a set of nucleotides that codes for the exact same protein. It might not have any effect at all. So mutations are kind of cool. They can have good effects, they can have bad effects, and they can have no effect at all. All right, this was our video for this week. A little note, this is my last week as your student teacher. So starting up next week, again, you'll have Mrs. Hughes once again as your teacher. I'll still be popping in and out doing some grading and things like that, but I just wanted to say thank you. You guys have been such awesome students. You've made my experience so great, so wonderful, and I'm so excited to be doing exactly what I love. Hopefully you've learned as much from me as I learned from you. And I just want you guys to keep being awesome, curious, fantastic people, because you guys are amazing.